2, and we're going to go through a lot of material today. Yes, we
Is
have
it
a question.
okay if I record the lectures?
Yes.
You meant? Okay.
Yes, absolutely. Did everybody download the PowerPoint from last week's lecture? You can found it online and everything. Okay. The other thing I wanted to mention was, did everybody download this document, Bluff Your Way Through Electronics? Okay. The next lecture, hopefully at the end of this lecture and the next 2 lectures, we're going to be talking about electrochemical gradients, Nernst equation, and all that. So it'd be really helpful if you kind of reviewed your electronics and your physics, especially if it was a couple of years ago. It's kind of helpful to do that. And so this is a really great guide that was written by Dr. Payne. How many of you have had Dr. Payne for a class before? Yeah. Yeah. So he's written a very nice guide. It's very, very useful. So take a look at that. And I also wanted to review TA and faculty office hours. In the syllabus, we've listed the TA hours. So Grace, stand up, wave, everybody. Grace will have TA hours prior to Thursday lecture in the grad offices over here. Chanmi, can you stand up and wave? Yep, just so you know faces. So she'll be available before Tuesday's lecture in one of the little seminar rooms right over here. I'm available on Wednesday afternoons from 2 to 4. And this is my office number, 4244. I'm in the 4th floor of the biology building here. Excuse me. If any of you need to talk to one of us and you're not available for these times, just shoot us an email and we'll come up with another time to talk to you, especially in the next few lectures. Often it's a little confusing, or you just need a little reinforcement to go over some of the electrochemical gradient stuff especially. So please use the office hours. We're happy to see you. We're kind of sad when nobody comes by. So kind of come on by. Okay, so I just wanted to kind of recap a little bit from last week's lecture. Oh yeah, that's just a reminder. Sorry. Here we go. Start over. Okay, I just wanted to recap last week's lecture, just to kind of give you an idea of, what is it that I want you to get out of the lecture. So I'll try to give you a little bit of a summary of what they call learning objectives for each of these lectures. I can get my laser pointer out of here. All right. So last week we talked about, sort of introduced neuroscience in general, and then we talked about the cellular structure of neurons. We talked a little bit about glia, what the different types of glia are and their basic function. Let's see, right thing. No, that's too dark. Which one is the right one? This one? Yes. Is that right? Yeah, that's good. Okay, and we reviewed a little bit of the history, so know who Ramon y Cajal is and what the neuron doctrine is, and the idea of dynamic polarization. How you could look at these circuits and kind of tell how is the information flowing in these circuits. And then we talked a little bit about neuroanatomical methods, and we kind of went over that quickly, so I wanted to give you a quick summary of the major methods, because we'll be looking at data from each of these methods as we go along, especially in this lecture. So a lot of what we know about brain structures is based on what's called the Nissl stain, and that gives you an idea of what the layers are, and it distinguishes nuclei. And it's basically based on the idea that neurons stain with this particular cytological stain, and it gives you a general layout. It tells you where neurons are more dense, where they fall into lines, and so on. We talked about the Golgi method. That was Camille Golgi who developed that method. Fluorescent dye fills, which is sort of a modern version of a Golgi method where you can dye individual neurons and you get a complete neuron. And you can use a microscopy called confocal microscopy to get a 3D image of these neurons. Immunostaining, and this is the technique where you use antibodies that recognize particular proteins, and then you tag them with either a fluorescent tag or an enzymatic tag that allows some sort of dark precipitate. And that gives you an idea of both the distribution of the particular protein of interest, but if you pick the right protein, it can tell you a lot about structure. We didn't talk a lot about track tracing, so this is a technique where you use either a fluorescent or radio-labeled dye, and then it's transported along pathways, so we can see sort of what's connected to what. We'll see a lot of that in the coming lectures. And then finally, we alluded to genetic encoding of fluorescent tags. So this is using molecular biology tricks where we know the gene of interest, and you can modify it by adding a fluorescent tag, such as green fluorescent protein, and then you can express it in these neurons. And the protein will go and mostly do whatever it is that it does, unless you tell it not to, give it a mutant version. But the fluorescent tag tells you exactly where those proteins have gone. So these are all sort of anatomical techniques that we'll use. And that tells us a lot about the nervous system. All right, so that was kind of basically the last lecture. Today we're going to go over gross anatomy and the neuroscience methods, and hopefully by the end of it we'll get into start into the electrochemical gradients, since we kind of lost a day at the very beginning. Okay, we already said that. Okay, so gross anatomy. So in your book, this is basically covered in chapter 1, but it's also a lot of information in the appendix. So go to the appendix and look at the pictures there, look at some of the descriptions. Basically what I'm going to ask you to cover is what we're going to cover in this lecture, rather than every detail in the appendix. So just use the appendix to supplement what we're going to talk about today. Okay, so basic structure of the brain and the nervous system. Mostly we're going to talk about the central nervous system, which is the brain and the spinal cord. People often confuse the spinal cord for peripheral, but the spinal cord is really part of the central nervous system. And then you have the peripheral nervous system, and this is everything outside of the brain and spinal cord, including sensory neurons, so like your somatosensory skin receptors and so on. And then a lot of the ganglia that sit outside of the spinal cord, and the visceral nerves. And we're probably not going to cover an awful lot of the peripheral nervous system. We'll be focused on the central nervous system. One caveat is that a notable exception is that the eye is considered part of the central nervous system. And this is because the eye and the optic nerve basically develop from the brain. So it's partly a developmental definition. Where do these structures come from? Okay, and so this is an illustration. So here we have brain and spinal cord. And the spinal nerves, and these are some of the ganglia that are outside of the central nervous system, the dorsal root ganglia, that are considered part of the peripheral nervous system. Okay, and so this is sort of what you should think of as the general layout. The brain is kind of a black box. You have sensory in and behavior out. And so the basic idea is that you have your external environment, sensory receptors that send sensory information to the central nervous system, cerebral hemispheres, and then those have a motor output, both the visceral and somatic motor system, that then is turned into behavior and movement through the muscles of your body. Okay, terminology. Terminology is really important because everything is going to be referred to by these types of terms. Okay. And so, what do these mean? So rostral, caudal, dorsal, ventral, anterior, posterior, superior, inferior. Some of these you can kind of guess at, but they have very specific meanings when we're talking about the brain. Okay, so let's talk about rostral, caudal, dorsal, ventral. And some of these terms kind of go together more than others. Okay, so in general, you want to kind of think of the brain. Let's look at the spinal cord. So the spinal cord runs from your head to your tail, okay? And so the tail is considered caudal, and then the head is considered rostral. Okay, so there's sort of this long axis. And if you imagine that most animals, humans are just a little bit weird, but most animals basically have a spinal cord that runs along their back, and then they have a head that looks forward, right? Like think of your cat, okay? So rostrocaudal is very obvious. You have the nose and you have the tail, okay? And then dorsal ventral is equally
intuitive, right? The dorsal is the back, ventral is the belly. Okay. Humans, on the other hand, are weird. We stood up and we tipped our head over. Okay, so this is what you need to remember when you're trying to figure this out, because now, when it comes to the brain, caudal is the back of the head, and rostral is your forehead or your nose. Okay, so there's a flexion in that long axis, and that affects what we call rostral and caudal. Does that make sense to everybody? It's probably the most confusing component. Okay, so rostral caudal, head to tail, dorsal ventral, top to bottom. Okay, and then these other terms are very commonly used, especially in neurology, and for naming brain structures. We have superior, which means top of the head, inferior, which generally means bottom of the head, maybe think of it under your chin, so top and bottom, and then anterior, front, posterior, back. Those are pretty intuitive. Okay. Oh no, let's move those around. Yes, I'm editing my slides as we speak. All right, that's better. All right, so this is just recapitulates what I just told you. And generally, you can think of these terms as pairs. So rostrocaudal are paired, dorsal ventral, they're opposites. And some of them go together better than others, like, and which ones we use are generally a matter of convention rather than logical consistency. Yes?
So in a human, is there much of a difference between rostrocaudal and anterior posterior?
It's mostly a matter of convention, but you'll see both terms used. Yeah. Okay. So often, nuclei in the brain are shown through using brain sections, right? You've all seen brain sections. Okay, and these brain sections have particular terms that you'll need to know. So the most intuitive is horizontal. So this is, and now we're just talking about the brain. We're not talking so much about the spinal cord. So you have a horizontal section that cuts straight through from front to back in the flat plane, horizontal plane. And then the two sort of specialized terms are coronal, which is, think of it as a crown, sort of, that cuts through here. It's also known as transverse. People refer to it as transverse. And then sagittal is from front to back, this way. So that sagittal splits the left and the right. Okay. And then we also have two terms, lateral and medial. You're probably familiar with this. Medial, meaning close to the midline, and lateral is to the side, right? So your ears are lateral. Okay, so let's look at sort of the basic structure of the brain. And there's a lot of terminology in this lecture, and I apologize. We'll kind of crank through it, and it's basically just going to be a matter of memorization. You've probably seen some of this before, but it's kind of, you can come up with mnemonics about how to figure this out. But you've probably seen a lot of this before. So this is basically a lateral view of the brain. So here's the anterior of the brain, or rostral. Here's posterior. Here's the cerebellum. That's always a giveaway. And so mostly what you see is the cerebral hemisphere. But as you can see, the human brain is highly folded, and it has many of these folds and outfoldings. So you can refer to those where it's folded out. Sort of the hills are the gyrus. And the infoldings are either a sulcus or a fissure. Okay, so a fissure is actually a complete separation of two brain areas, whereas a sulcus is just an infolding that's eventually going to come back out. Okay, let's back up. Let me notice one more thing. So there are particular sulci and gyri that are important. One is the central sulcus. It's kind of here. Just in front of the central sulcus is the precentral gyrus and the postcentral gyrus. So before and after the central sulcus. Let's see, here's the lateral fissure that separates the frontal lobe from the temporal lobe. I think that's supposed to talk about. And these different divisions roughly divide the cortex into these different areas that you're probably somewhat familiar with. So there's the frontal lobe. The central sulcus divides the frontal lobe from what we call the parietal lobe. So here's the pre-central sulcus, and here's pre-central gyrus and the post-central gyrus. Okay. Here's the temporal lobe. Here's that lateral fissure. And then the occipital lobe sits on the very back. The occipital, we call the occipital pole, the very posterior pole of the cortex. Okay, so this is a lateral view. This is what we would call a medial view. So this is what we would call basically a mid-sagittal section. So it's basically like you just cut the brain in half and you're looking at the inside of it, the medial face of it. And mostly what you can see here, you see these are the cortical lobes. This structure right here is the corpus callosum. That'll be in another diagram, so don't worry about labeling it. But you can see that these lobes kind of are mostly defined in sort of an anterior-posterior way. Okay, so each of these lobes are basically subserve different functions. Okay, so the occipital lobe, you're probably already somewhat familiar with, subserves vision. So both your primary visual areas and some secondary visual areas. The parietal lobe is involved in somatosensation and other things, but for now, this is what you need to know. Frontal lobe, lots of complicated things, but you could think of it as the motor planning area. Some people talk about it as an executive function area, sort of higher cognition area, and so on. But most especially, it's the motor planning area. And then the temporal lobe is involved in hearing, but as you'll see, a lot of the medial temporal lobe is involved in higher functions such as memory, that we'll talk about a lot later in the course. Okay, so here's a number of these things labeled. So this is a dorsal view of the brain. Anterior is to the top, posterior is to the bottom. Here's a ventral view. Okay, here's the longitudinal fissure that separates the left and the right halves of the cortex. Here's your central sulcus. One notable thing is that the left and the right are not perfectly symmetrical. Everybody's also a little bit different, so you have to take a particular picture of the human brain with a grain of salt. They're always a little bit different. If you cut away sort of scoop away the very top of the brain here, you'll reveal the corpus callosum. So that's an area of white matter that connects the left and the right hemispheres, sort of the superhighway of the cortical brain. Okay, and then here's a dorsal view of the frontal lobe, parietal lobe, occipital lobe, and then in the ventral view, you can see the temporal lobe here and some of the frontal lobe. On the ventral view, there are a number of other structures that are visible. There's some sensory structures, such as the olfactory bulb, that connects to olfactory cortex via the olfactory tract. Here's the optic chiasm. This is where the optic nerve then branches out and goes out to the eyes. What else do I want you to know? You can see the cerebellum. And then here you have brainstem. You get a very good view of the pons of the brainstem. And then here you branch off into the spinal cord. Are there any questions so far? Yes.
So we should know, as far as the underside of the brain, we should be able to identify the olfactory bulb, optic chiasm, and then cerebellum and pons.
Yeah, and I think just generally understanding that you can see the temporal lobe here.
Yeah, okay.
Yeah. But basically consider anything on these particular slides fair game.
Okay.
But those are the most important structures. Okay, so just to kind of summarize, from a surface view, you have the left and right hemispheres divided by the longitudinal fissure. You have gyrus and sulcus, which are the infoldings and outfoldings of the brain. The most critical is central sulcus. We're going to come back to that a lot. And so you can see that mostly the cerebral cortex, the cerebellum is visible, the brainstem, and a few other landmark structures that you can see on the ventral view. Okay. So this is another view of the mid-sagittal section. So now we're looking at the medial surface. We just talked about the cortex and the corpus callosum. But in this section, you can also see several other structures that you should take some time to learn. This is the diencephalon. That contains the thalamus. Then you have just below the diencephalon. This is often called, this is the diencephalon and the cortex together are often referred to as the telencephalon. That'll be on a later slide, so you don't have to write that down right now. But the telencephalon and the diencephalon together generally are considered forebrain. Okay, and then below that is the midbrain. And below that is the pons and the medulla, which together are often referred to as hindbrain. But this entire structure, a lot of people refer to as brainstem. People like me who work in hearing kind of consider brainstem to be everything but the midbrain, but for simplicity, we'll just refer to how the book refers to it here as the brainstem. Okay, so these are the major structures. Brainstem, the brainstem is really, really critical. You know, if you have brainstem damage, it's a bad thing because that controls your respiratory centers, often controls components of movement. It's composed of the pons, medulla, and the midbrain, and it's the target of a number of cranial nerves, which we'll get to in a minute. Then you have cerebellum. Diencephalon consists of the thalamus and the hypothalamus, which have very important functions. The thalamus in particular is sort of a hub of sensory information processing. Everything comes up kind of through the thalamus and then out to the cortex. Hypothalamus, you've probably heard of before, is really important for hormonal control and different aspects of appetitive behavior and so on. The telencephalon is composed of cerebral cortex, the cerebral hemispheres, corpus callosum, and various tracts of white matter. So forebrain is generally the diencephalon and telencephalon together. What we term the hindbrain is the pons and medulla, and then you have the midbrain. Okay. So some of these terms are sort of overlapping in what they refer to, and that's because people have been studying different aspects of the brain and in different animals. So I apologize if it's a little bit confusing. It's sort of part and parcel of neuroscience, I'm afraid. Okay, so there are other structures. So that's sort of these superficial structures that you can see from the outside, but there's a lot of stuff going on the inside, obviously. And one of the major structures, so we're going to start cutting through the brain. So here's a lateral view, and we're going to look at two sections, coronal sections, one at this level through the brain and another at this level. And on the inside, what you're going to see is that it cuts through these interior structures that are referred to generally as the basal ganglia. Okay, and that's composed of the putamen and the caudate. And then there's the amygdala down here, and then this little purple region here is the diencephalon thalamus. The thalamus of the diencephalon. Okay, so first we're going to look at through this section. And this is what a typical sort of coronal section is. So you're cutting straight down through the head like this. Okay, so just to orient you, here's the midline. This is the dorsal surface. Here's the ventral surface. And of course this is medial, lateral, lateral. Okay, and it's pretty symmetrical. So you have, the most notable thing here is that you see the gray matter. You see the folds of the cortex, and you see that there's what we call gray matter. It looks very yellow here, but it's gray matter. And then underneath the gray matter, you have the white matter. You guys know what the white matter is?
Axons. Axons.
Axons. So the gray matter is generally cell bodies, dendrites, neuropil, synapses. And then the white matter are the highways of axons that connect all of these different parts. So every part of the cortex has to get inputs from somewhere and it has to send outputs. Okay, so that's what all of the white matter is. Let's see. So that's the cortex. You have temporal lobe, basically frontal lobe here. And then in the interior, here you have the corpus callosum. So that's that big superhighway of white matter that connects the two hemispheres. And then you have these structures. And so these structures, they have that three-dimensional space-filling model. Let's go back. Okay, so you have to think of that structure. And then think of this as a cross-section through that structure. So here is the caudate. Here's this triangle of gray matter is the putamen. You have another layer of white matter, another little highway that runs here through the anterior commissure. And then there's another nucleus called the basal forebrain. And then the amygdala is sitting down here. So you'll have to kind of work through the concept of what a cross-section through the brain looks like. Now, if we move a little bit caudal and cut another cross-section, it looks a little bit different. We have some similar structures, so we still have, the cortical lobes. We still have the corpus callosum. But now, let's see if I can see this. The caudate is just this little section right here. The putamen is over here. Now there's a new structure called the globus pallidus, which is also part of the basal ganglia. And now you've got a big section of the thalamus. So this entire round section here, which itself can be divided into these little subsections, which we'll talk about when we get to each of those a little bit later. Okay. You can also see that there are these holes in the brain at various areas. Those are the ventricles. Yes?
I just have a small question about the placement of the amygdala. Is it only on the left side of the brain?
No, it's symmetrical. Yeah, so a lot of these labels are only labeled on one side, and you can assume that there's a symmetrical structure on the other side. I think these are very good sections. Let's see. For example, here is hippocampus, and then there's a similar structure on this side, but they're not exactly, precisely symmetrical. But wherever there's a structure on the left, there's also a structure on the right. Nothing's sort of missing. We'll talk about laterality later on, but it's not at this level of structure. Any other questions? Yes?
Are these cross sections are you looking at from the front or the back?
Oh. Do you think it makes a difference?
I mean, some of the structures.
Sorry, the question was, are you looking at this from the front or the back? Does it make a difference? Yes. How many yeses? How many no's? What?
It's parallel, so it doesn't make
Yeah. The answer is it depends. It depends on how thick your section is. So if you have a very thin section, it basically looks the same from the front to the back. If you have a very thick section, you might catch different structures on the front and the back. So that's a good question. So it depends on how thick the structures are. Yeah. I think you can assume from this that we have it looks thick in this photo, but almost like we just sort of cut away the front of it. So I would assume that you're looking nose on, if it helps you. In this particular, it probably doesn't matter, but if you are doing the sectioning, it matters a great deal. All right, what else do we need to see here? So, ventricles. Anybody know what the ventricles are for? What fills the ventricles? Cerebral spinal fluid. Right. So what's important about cerebral spinal fluid?
I mean, it's what the brain basically floats in and it cleans it out and brings nutrients in.
Right. So it brings nutrients, it removes bad things, it cushions the brain. Yeah. All right. Okay, let's kind of speed along. So this just kind of summarize what we're looking at. So this is, so now we're going to go sort of below the forebrain a little bit. And this is a ventral view of the diencephalon, meaning the thalamus, the midbrain, which is located here, and then the rest of the brainstem. And one important thing to remember, this is a ventral view, and we have taken away the cerebellum. We're not going to talk about the cerebellum today. That's going to be a later lecture. Just to, but if you cut away the cerebellum, so you make a cut right here and right here. These are the cerebellar peduncles. So there's a position, a place here in the pons where you have all of the fiber tracts that go to the cerebellum. Okay, you cut those away, and you're left with the brainstem here, the pons, medulla, down to the spinal cord. Okay, this is sort of the same ventral view. So here's your brainstem, but now we've left the. Sorry, this was a dorsal view. This is a ventral view. So now we've turned it over, and this is the brainstem. Here's the cerebellum, and so this is a view that shows you the cranial nerves. This is, I think this might be in the appendix. Is it? Yes. Okay. Yes, this is in the appendix. And you don't necessarily have to remember all of the cranial nerves, but there are very key ones that we're going to talk about in later lectures. When I took physiological psychology in college, I had to memorize all of them. And if you Google cranial nerves mnemonics, people have songs and all kinds of ways to remember this. So it's very useful. But the ones that I really want you to note are some of the sensory inputs, the olfactory nerve, which is really a tract here. It's not sort of a thin nerve the way we usually think about it. Number 2 is the optic nerve, the oculomotor nerve. Number 5, the trigeminal nerve, the vestibular cochlear or auditory nerve, that's kind of my favorite, and the vagus nerve, all things that are going to come up in later lectures. But this is a mapping of all of those nerves. So those ones that I've highlighted are the ones you should try to remember. Okay, and then this just summarizes what those are. I'm not going to go over that. Okay, so the basic idea here is that I want you to get familiar with and be able to use the terminology that we use to label direction and orientation of these structures. You need to name and identify some of these major brain divisions. Name and locate the cortical divisions, frontal lobe, temporal lobe, and so on, and what their sort of general function is. And name and locate some of the hallmark structures in the sectioned anatomical figures. So just focus on what we covered in the lecture today, and you'll do all right. All right, moving on. So in this course, we're going to really focus on neuron as kind of the functional unit of information processing in the brain. Obviously, the brain is composed of neurons, but individual neurons are exquisitely set up in order to process electrical activity. So their properties, along with their connectivity, is really what makes the brain do what the brain does. Okay, so today we're going to talk about signaling via action potentials within a simple circuit. And then hopefully we'll get to some of the methods involved in understanding the functional analysis. We really think of this physiological activity in the brain. Okay, oftentimes the brain is compared to a computer. So in what ways is it sort of similar to a computer? So one of the ways that we can think about this is we can ask whether the brain is analog or digital. So we all think of digital computers, but of course there are analog versions of computers as well, analog circuits. But generally the idea is that analog is a graded signal, and digital is in bits, 0 or 1, all or none. And one of the advantages of using digital is it gives you a clean signal. It's either there or it's not. And so it gives you a very nice way to signal information. And then of course you can combine bits to create basically any kind of information. And the way that we're going to talk about this is as a computational unit at the level of the neuron. Okay, so electrical information. So the brain is an electrical device, essentially. So neurons convey electrical information via changes in the plasma membrane potential. Okay, so all cells generally have some sort of potential, but neurons have membrane potential, and they have active membrane potential. So the electrical potential difference, there's a difference between the inside and the outside, and this membrane potential can flow. So it can move from those dendrites down through the axons, just like Cajal envisioned in his anatomical slides. Okay, but basically these kind of changes in membrane potential can take two forms. So they can be sent as action potentials, which you can think of it as digital, or similar to a digital signal, and graded potentials, which are similar to an analog signal. So it's a little bit of both. So action potentials, sorry, I'll refer to them colloquially as spikes. You'll hear me say that a lot more than action potentials because it's a lot quicker to say. And so the advantage is that these can really travel long distances. It's usually a big signal, and it can travel down the axons without losing any amplitude. And we'll talk about specialized ways that that amplitude is maintained. It's generally a fixed amplitude, so we refer to these as an all or nothing. And it's a very short duration, generally 1 to 10 milliseconds, depending on the animal and the temperature and so on, but very short. So it's a blip, right? A spike. These are triggered in the axon or cell body, and really they depend on graded potentials in order to get going. Okay, so a graded potential, this analog signal varies in amplitude. It could be very small, could be very big, or any shade in between. Okay, graded potentials, when we talk about them, these are largely confined to the dendrites, the cell body, and sometimes very short axons. They don't travel more, they tend to decay, so they're not as reliable as an action potential. But the amplitude is related to the intensity of the event which initiated them, which in some cases means how much neurotransmitter was released, or how much pressure was applied to a sensory receptor. So in that case, the analog signal is basically a direct readout of the input that it's getting. And these are relatively longer duration, of 10 to hundreds of milliseconds. And this allows a neuron to basically integrate information across time. All right, so let's look at this in terms of a very simple circuit. Okay, this is a simple knee-jerk response, the knee-jerk reflex. All of you have had this done to you at the doctor's, where they apply a stimulus to your knee, and then your foot is supposed to kick up a little bit, right? Okay, and so this is basically, sort of the canonical example of a simple reflex circuit. Okay, so what's happening here? Okay, when the hammer stretches your tendon, there's a sensory receptor in the muscle spindle that sends a signal to your spinal cord. And then in the spinal cord, it contacts two other neurons. So there are two neurons. One is a motor neuron. So let's look at the direct pathway. So it contacts the motor neuron, it stimulates the motor neuron, which sends a signal back to the muscle, causing the muscle to contract and your leg to expand. Making this slightly more interesting is the fact that you have muscles that work against each other. Okay, so you have a second contact. So the sensory neuron also contacts an interneuron in the spinal cord, which in turn inhibits a motor neuron, the second motor neuron, that then goes to your hamstring. And that allows that flexor muscle to relax, and so it's not going to oppose the extensor muscle. Right? Very simple. Okay, so this is basically what I just said. Okay, so what happens here is the sensory receptor receives a graded input and then sends an action potential. That action potential releases neurotransmitter, causing a graded input to the motor neuron, which then transforms it into an action potential, which then provides neurotransmitter to the muscle, which causes the muscle to contract. Okay, so this is all that in words. Okay, so how are we going to show that? Okay, so typically we know all of this through various forms of electrophysiology. And sort of the most fundamental form of electrophysiology is by putting an electrode near where the source of the activity is. So in this case, this is actually a slice through a cortex, and you have different neurons, and you put an electrode near to the neuron that you're interested in. And it picks up extracellular signals. Okay, and so we'll talk a little bit about the generation of those signals, but basically consider that when there's a change inside the cell, there also has to be a change outside the cell. Okay, voltage potential is a difference between the inside and the outside. And what you're picking up with this electrode are these spikes of changes in the voltage. Okay, so what does that look like in this circuit? Okay, so in this circuit, so here we're going to show the spike activity, which is often illustrated in this manner, where you have time along the x-axis, and then a blip in that time is an action potential. So this is the electrode picking up an action potential. So this is a sensory neuron, and then we'll have a separate electrode that's recording from the motor neuron, say, another electrode that's recording from the interneuron, and another electrode that's recording from the motor neuron. So what's kind of critical here is that each of those electrodes is picking up a single neuron, okay? So when we have the hammer tap, the sensory neuron, usually the cells often have some sort of baseline spiking. They're just kind of popping away. But when you have a sensory stimulus, you get an increase in firing in the sensory neuron. That releases neurotransmitter onto the motor neuron, and so with some delay, you get an increase in firing in the motor neuron. So there are more action potentials, and an increase in firing in that interneuron, because the sensory neuron is releasing neurotransmitter onto both of those at the same time. Now the interneuron is contacting the flexor motor neuron, but look what happens, is you have a break in the firing. So there's some sort of baseline firing, and then it stops. Okay, why is that happening? Okay, to look at why that's happening, you get basically an inverse signal. We're going to look at the intracellular potential. Okay, so extracellular recording tells you about action potentials, but it doesn't give you any information about what we were calling the graded potentials. So intracellular recording, and we'll talk a little bit more about the methods, but it's basically taking something like a glass pipette and either sticking it inside the membrane or patching it onto the membrane and rupturing the cell membrane so that you have electrical access to the inside of the cell. So now you're measuring the inside relative to the outside, whereas the extracellular potential is measuring the outside relative to some sort of faraway ground. Okay, so this is an example of the sensory neuron, an intracellular recording from the sensory neuron. The illustration here, it looks like you're recording from the cell body, but to be a little bit pedantic, you actually would need to be recording from the dendrite where the graded potentials are happening. Okay, so this is a sensory neuron recording in the dendrite. And on the left, you have membrane potential, and on the bottom, you have time. So at the time that you tap the tendon with the little hammer, the membrane potential shows an increase or depolarization in the membrane potential, and that's that graded potential. So those are the sensory receptors in that neuron activating. And we'll talk ad nauseum about how these things happen, but just for now, it evokes a graded potential, and at some threshold, you get an action potential. So this is what we call a spike threshold, okay? And that's basically the threshold at which you get that all or none signal. So the spike in the voltage goes up and it goes down and has this sort of characteristic shape, and then it goes back to baseline. So graded potential, action potential, okay? The sensory neuron then contacts the motor neuron. The extensor motor neuron is shown in this panel right here. So you have the baseline membrane potential, and then when the sensory neuron releases neurotransmitter, it has a graded potential that then reaches spike threshold, and then it has an action potential. It's kind of cut off a little bit here, and it goes back down. Okay, and two things are shown here. One is shown the graded potential if a spike had not happened. Basically, it goes up, then it goes down. It has this kind of longish time constant to it. But if it reaches threshold, then it's superseded by the action potential spike. In the interneuron, we have a similar thing happening because the sensory neuron basically has the same effect on both of those. You have a graded potential. If it reaches threshold, it fires a spike. Now the interneuron also releases neurotransmitter onto this flexor motor neuron, but the transmitter has an opposite effect, right? And we'll talk a lot about excitatory versus inhibitory. So if the graded potential goes up, we call it excitatory. If it causes the membrane potential to go down or hyperpolarizes, we call it inhibitory. And the reason for that is because if you push the membrane potential down, it becomes even less likely to fire an action potential. Right? It's very intuitive. And then it comes back. Okay? So this explains this activity here. Right? So the sensory neuron is causing a graded potential and firing in the motor and interneuron. The interneuron, in turn, is causing a hyperpolarized inhibitory potential in the motor neuron, and so it shuts off. All right, so to summarize, information is encoded in the dendrites and at the cell body by summing up these graded potentials, either through the sensory inputs or through the neurotransmitter. Larger the stimulus, the larger the graded potential. In the axons, you have this threshold mechanism, and where you get either an all or none signal. So either you don't have enough input, there's not enough of a graded potential to fire the cell, to fire an action potential, and it just kind of fades away, or you reach threshold and it fires an action potential. And if you have lots of graded potentials continuously coming in, it will fire multiple action potentials. Okay, so dendrites basically function as an AM signal, an amplitude modulated signal. And another way to think of it is the axons function as a frequency modulated signal. So in the dendrites, you're counting the amplitude of the graded signals. In the axons, you're really counting how many spikes do you have. So a spike count. All right, we're going to kind of run out of time, so we're going to have to kind of continue on. Let's see, comparisons with computers, interesting but not that critical. Okay. So in a lot of cases, like the thing that we just talked about, the reflex that we just talked about, where there was a decrease in firing, that was because there's often some sort of baseline firing level. So most neurons are kind of popping away at some baseline level. And in order to get your hamstring to relax, you actually have to decrease the firing. So there was some basal firing, right? Because when you're sort of standing, you're not running, but you have to have some sort of baseline level of tone to your muscles, right? And that's because your motor neurons are kind of continuously firing. Okay, so sometimes information is because you have an increase in the firing rate, and sometimes the information is because you have a decrease in the firing rate. So both directions are important. All right, so we talked about measuring electrical signaling, intracellular and extracellular signaling. We're going to start to talk a little bit about various methods. So these electrophysiological methods, you're recording the voltage directly, but there are a lot of ways to sort of get an indirect measure of voltage activity. You can use a dye that's sensitive to voltage, or you can use a dye that's sensitive to calcium levels. So calcium often rushes into neurons, and so that's correlated with a level of changes in the voltage, but it's indirect. Okay, we talked a little bit about intracellular recordings, so this is an example. Intracellular recordings are generally performed using microelectrodes, like small glass pipettes that either penetrate the neuron or patch onto the neuron. All right, we're going to talk about different kinds of graded potentials. Some of them are called sensory generator potential. That's due to the sensory apparatus. We'll talk a lot about that. We're going to talk a lot about synaptic potentials. And so these are the potentials that arise with neurotransmitters and ion channels that are sensitive to those neurotransmitters. Okay. What we just talked about mostly was recording from single neurons, but there are a number of methods to record from multiple neurons. One of those is a multi-electrode array. So this is an electrode. This is basically a metal probe that then has a number of electrodes etched into the metal. And so it's going to basically sample the voltage at different sites. And so when you have a cortical layer like this, where you have a large number of neurons, you can sample from multiple layers. And there are methods to differentiate the signals in that system. This is an example from talking about calcium imaging. So you apply a dye, a fluorescing calcium dye, and then you can detect changes in the fluorescence. One of the drawbacks to using a calcium dye is that it's relatively slow. And it's relatively diffuse. But methods are getting better and better to record from whole areas of neurons. So this is an example of using calcium dyes in an olfactory system. So this is, let's see, which one is this? This is the olfactory bulb. And so from different areas in the olfactory bulb, olfactory bulb, where you have these layers, where you have the inputs, and then you apply a particular sensory input, and you measure the changes in the calcium levels. Okay, there are some more additional methods that are being used now. So instead of applying a calcium dye that's taken up by all of the cells in an area, is you can direct calcium dye using the miracles of genetics. So there's a particular calcium imaging technique called GCAM, which is a genetically engineered calcium detector, calcium dye, that is directed into particular cells or particular areas. And so now you can have genetically engineered animals that basically fluoresce their calcium signals in a very particular way. And people are starting to be able to do this in whole animal structures. So we've been able to do calcium imaging in live in vivo animals for a while, but now people are starting to use this in animals that are behaving, moving, which is really cool. And so there's a methods paper and a video that I think we're going to skip in the interest of time. Additional methods involve a technique called optogenetics. So in a sense, GCAMP is kind of optogenetics, but this is a particular, this really refers to a particular type of methodology where you're using not a simple calcium sensor, but a sensor that then senses light that you as the experimenter apply. And in this way, you can start to not only record from neurons, you can control them as well. And there's some really terrific YouTube videos that describe these techniques that we don't really have time to do today. But the basic idea is that people discovered these light-sensitive proteins that were originally from algae. And people have made all kinds of variations on this. But the idea, like green fluorescent protein, is that you can encode it, take the gene for that protein, and insert that DNA in particular neurons. And so you can select promoters, so you can express it in particular subsets of cell types in the brain. Okay, this is a protein that responds. It's an ion channel, and so it responds when you shine a blue light on it. And so the experimenter can take a little blue laser, and when you apply it, you will open the channels. And so what happens is that any neuron that's expressing this channel will now either depolarize or hyperpolarize, depending on which version of the protein that you have expressed in these cells. And so this is a methodology to, meticulously, carefully control cellular activity at the cellular level in the brain. Okay, there's a number of systems. This is from Drosophila. So there's a whole range of Drosophila that have very specific genetically encoded sensors and reporters, different reporter lines. And so you can take identified neurons, so we know basically every neuron in the fly brain and what promoters are involved. And so you can very specifically express different types of dyes. And so this is just several different identifiable neurons. People are starting to do this in the cortical system. It's much more complicated in the cortex. You don't have identifiable neurons like you do in the invertebrate system, but it's starting to work. This is just sort of this is more of a demonstration of principle where you can use multiple colors in order to differentiate different cells that are right next to each other. And I think in the last 15 minutes here, we're going to move on to some basic non-invasive methods. So how do you study things in humans? Okay, because you can't, except for in the case of a few epilepsy patients, you really can't do invasive procedures in most humans. Okay, so we'll talk for a few minutes about these non-invasive techniques. And you've probably seen all of these, right? So probably very common is CT scans and PET scans that are often used in the medical field for diagnostic purposes. And we're not going to talk too much about that. And then a couple of other very important techniques, especially in research, are EEGs, although this is used a lot in sleep research, MEGs, and different forms of MRIs. So CT scans, what I really want you to know out of here is the difference between each of these types of non-invasive techniques. You know, what are the advantages and disadvantages of these different techniques, and sort of the basis of why they work. Not in too much detail, but sort of the basics. So a CT scan is based on X-rays. So it's basically just a very narrow band X-rays, and it gives you a structural idea of the brain. So it differentiates between gray matter and white matter, and so it's a very good diagnostic tool. Make sure I'm on the site. Okay. EEGs are something that's probably also very familiar to you. So this is where you put on that array of electrodes, often in a little cap. And so it's a direct measurement of electrical potentials in the brain. So you have this sort of suite of EEG traces. And so you get very good spatial positioning. So it's at very particular points of the cap. And you get very good temporal signal here. Let's see. So you have arrays of electrodes. You're directly detecting electrical potential. Can be used to determine sleep-wake states, REM, REM sleep, and seizure activity in particular. Okay, one thing that we don't know very well is exactly where these signals come from, right? So you're detecting electrical fields, but electrical fields are not very precisely localized. They emanate out. They have sources and sinks, so it's not entirely clear where these signals are coming from. But one thing that you do get is you get very good temporal resolution. So here you have time on the bottom axis, and this is just an example of different qualitative differences in the activity of EEGs. Okay, and so these have been used for decades and decades, so people have a pretty good idea of what they're looking for. Another technique is MEG, magnetoencephalography. So this is sort of a flip technique. EEGs detect electrical activity. In this case, you're using a device called a squid device to detect the tiny magnetic fields that are generated by the electrical currents in the brain. So those electrical currents that you're detecting by EEGs also emanate magnetic fields, and they're teeny, teeny, tiny. And so this is a way that you can basically detect those fields. It also has a very good time resolution and relative spatial information, and it's a relatively quiet technology, unlike MRIs, which are very, very loud. Okay, so this is often used in hearing research because people can actually hear pretty well. And then also familiar to a lot of you is MRIs. So the basic MRI is detected by using very strong magnets. So if any of you have had an MRI, people have had to make sure that you're not wearing any jewelry, you don't have a pacemaker, you don't have a cochlear implant, you don't have a plate anywhere, screws in your knees, those sorts of things, because it uses such high magnets that basically kind of rip things off of your body. Not good. And so the basic methodology, you don't have to understand the physics of it here, but the basic idea is that it detects, it sets up a field that allows the hydrogen nuclei to orient in this magnetic field. And then it uses an RF pulse to disrupt some of these hydrogens, and then it detects the energy that is a result of the interaction between the magnetic field and the RF pulse. And the basic idea is that then you're detecting those energy emissions at a very high resolution, relative to the CAT scan and so on. And this is anatomical. So you're getting basically a static picture. And it really depends on the amount of water in different components of the brain. So a lot of those membranes, remember, are very fatty, right? And so you have varying amounts of water and fat in various structures of the brain. Okay, but this is an anatomical method. It's a static method. So this is just a picture of the MRI scan. This is an example of both an MRI and an fMRI. But this will give you an idea of sort of the quality of the images. So you can take, stacks of images. But you can also use a functional aspect of this. So somebody discovered that the amount of oxygenation in the blood can alter how these magnetic fields are affecting the hydrogen in the water. Okay, so the hemoglobin is going to distort the MRI resonance, and that's going to change the signal. And so you can detect those changes in signal. So you basically get a baseline scan, and then you get a scan of somebody doing something. And what it shows is a difference in blood flow. And so the idea is that the blood that's flowing to parts of your brain that are being more active. You're probably all really familiar with this already. And so basically fMRI depends on blood flow, which is an indirect measure of electrical activity. Okay, but it's a more dynamic measure. Okay, so you get a spatially accurate image. It's relatively slow because you have to take a lot of scans over time, but it gives you an idea of what the activity is like in the brain. And so this is a scan. So this is actually both a standard MRI and an fMRI. So this is an example of a patient with a tumor. So this white structure here is the tumor. And then this person is doing basically a manual task. And then the colored images are the differences in the blood flow while the person is doing a task, either on the left or on the right. Okay, so that gives you an idea of where different structures are and how it might be affecting activity. And of course, also people often do this in research with normal patients, non-pathological patients that are just doing some particular task. All right. So then there's one more variation on the MRI called diffusion tensor imaging. And the basic idea here is that I don't understand the physics of it, but the basic idea is that it's detecting the flow of water, the diffusion of water. And the idea is that when you have fiber tracks, that's going to restrict the flow of water in one direction or the other. And so you can actually follow fiber tracks throughout the brain. This is kind of a flat image, but you should go onto YouTube and look at some of the, do I have one? Oh yes, here's one here. So you should go ahead and take a look at that and see it in 3D. It's really, really cool. And the basic idea is that it's a non-invasive way to do track tracing, essentially. And you can measure the connections between different places. All right. So one of the other aspects of sort of modern neuroscience are sort of this sort of big data approach to neuroscience, the idea that you want to map every connection in the brain. So the diffusion tensor methodology is to measure every connection like this in the brain, in the whole brain. And then there's efforts underway to basically model the entire brain and activity in it. And I have there's a couple of links here, so you can go ahead and take a look at that. But I want to sort of close out today with a particular example of the idea of the connectome. How many of you people have you heard of something named a connectome? Sort of, raise your hand all the way. All right, sort of. Okay. So this is kind of one of the big things in neuroscience right now. So the basic idea is that if we could get a reconstruction of every cell and every synapse contacting every other synapse, then we could really learn something about the brain. Okay. There are proponents and there are skeptics, but it's kind of a cool idea. And the basic idea behind this version of the connectome is to get a 3D large-scale reconstruction of the entire brain. Okay, and here is a little cube. This is, maybe, I don't know, less than a millimeter by a millimeter section of rat cortex. Probably smaller than that, since I think these are cell bodies. And so all of these colors are all of these different components. And the idea is that you can do electron microscopy reconstruction. So electron microscopy is where you get really thin sections, and then you shoot electrons through it to look at the ultrastructure. Have you guys heard of ultrastructure, electron microscopy? This sort of thing. It's basically in order to get a spatial resolution that is way beyond light microscopy. So it's at the level of electrons. And so you can see synapses, and you can see endoplasmic reticulum and those sorts of structures when you manage it this way. So the idea is that you can do EM reconstruction of slice after slice after slice and reconstruct in 3D what the entire ultrastructure is like. So I'm going to play this for you because it's just kind of cool. And why not? All right. All right, so this is, I'm not actually that big a fan of TED Talks. I've included a lot of sort of links to TED Talks. You should be skeptical of a lot of the TED Talks that are out there, especially about neuroscience, but this is kind of a good one. This is Sebastian Sung, who is it MIT or Geneva, I can't remember. Right.
Now, you've probably seen pictures of neurons before. You can recognize them instantly by their fantastic shapes. They extend long and delicate branches, and in short, they look like trees. But this is just a single neuron. In order to find connectomes, we have to see all the neurons at the same time. So let's meet Bobby Kasturi, who works in the laboratory of Jeff Lichtman at Harvard University. Bobby is holding fantastically thin slices of a mouse brain, and we're zooming in by a factor of 100,000 times to obtain the resolution so that we can see the branches of neurons all at the same time. Except you still may not really recognize them, and that's because we have to work in 3 dimensions. If we take many images of many slices of the brain and stack them up, we get a 3-dimensional image. And still you may not see the branches, so we start at the top, and we color in the cross-section of one branch in red. And we do that for the next slice, and for the next slice. And we keep on doing that, slice after slice. If we continue through the entire stack, we can reconstruct the 3-dimensional shape of a small fragment of a branch of a neuron. And we can do that for another neuron in green. And you can see that the green neuron touches the red neuron at 2 locations. And these are what are called synapses. Let's zoom in on one synapse and keep your eyes on the interior of the green neuron. You should see small circles. These are called vesicles. They contain a molecule known as a neurotransmitter. And so when the green neuron wants to communicate, wants to send a message to the red neuron, it spits out a neurotransmitter. At the synapse, the 2 neurons are said to be connected, like 2 friends talking on the telephone. So you've seen how to find a synapse. How can we find an entire connectome? Well, we take this 3-dimensional stack of images and treat it as a gigantic 3-dimensional coloring book. We color every neuron in a different color, and then we look through all of the images, find the synapses, and note the colors of the 2 neurons involved in each synapse. If we can do that throughout all the images, we could find a connectome. Now, at this point, you've learned the basics of neurons and synapses, and so I think we're ready to tackle one of the most important questions in neuroscience. How are the brains of men and women different? According to this self-help book, guys' brains are like waffles. They keep their lives compartmentalized in boxes. Girls' brains are like spaghetti. Everything in their life is connected to everything else.
Okay, just ignore this part. We're
You
just
guys
going to
are
get on
laughing,
to the
but
next part.
this book changed my life. But seriously, what's wrong with this? You already know enough to tell me. What's wrong with this statement? It doesn't matter whether you're a guy or girl. Everyone's brains are like spaghetti. Or maybe really, really fine capellini with branches. Just as one strand of spaghetti contacts many other strands on your plate, one neuron touches many other neurons through their entangled branches. One neuron can be connected to so many other neurons because there can be synapses at these points of contact. By now, you might have sort of lost perspective on how large this cube of brain tissue actually is. And so let's do a series of comparisons to show you. I'll assure you this is very tiny. It's just 6 microns on a side. So here's how it stacks up against an entire neuron. And you can tell that really only the smallest fragments of branches are contained inside this cube. And a neuron, well, that's smaller than brain. And that's just a mouse brain. It's a lot smaller than a human brain. So when I show my friends this, sometimes they've told me, you know, Sebastian, you should just give up. Neuroscience is hopeless.
It's hopeless.
Because if you look at a brain with your naked eye, you don't really see how complex it is. But when you use a microscope, finally the hidden...
All right, so. Oh, don't do that. All right, so you get an idea of the daunting challenge there. All right, so. Okay, so what do I need you to get from this lecture about methods? We went through a lot of different things. Okay, and so the basic idea is that you need to understand how experimental design determines which method you're going to choose. Okay, are you trying to record from single cells, multiple cells, or from a large brain area or whole brain? What time scale are you trying to get information? What spatial scale? Do you need an invasive technique or a non-invasive technique? What's the physiological basis for the method? Is it a direct electrical detection, or does it detect indirect things like blood flow, calcium level, or maybe the tissue composition? For the human imaging, try to think about which of these methods are static versus dynamic, which gives you either anatomical, structural detail, or functional detail. Okay, so that's the kind of thing that you might be asked is, here's an experiment, what kind of method are you going to use in order to get the information? Okay, so that's it for today. So next time, voltage, current, and resistance.